see. Of course, I don't see it. Okay. Oh. So we'll wait for them to come back. And I have it all set up. So I'm going to ask them if they want to start with an overview of sequencing or walking through the Patrick assignment. Perfect.
I thought the PowerPoint that went through the Patrick assignment was like really good. Like it went through every individual step.
Thank you.
That's nice feedback to hear. Way to go, Abby.
I think way to go, Amanda.
Yeah, like my school, like we don't really do that many labs. Our science department is very small. So that was like my favorite lab I've ever done. Like actually seeing something happen, like seeing the mutations, it was really cool for me.
Oh, that's awesome. I got really nice feedback from you in the little Google Classroom thing, and I was thinking I wanted to let you know too, if you're interested, we can email each other, but we also, and for anybody actually as people are joining, but we have kind of a beta version of another way to analyze the sequencing data that it's nice because the output is way more visual, like to see the mutations. So maybe if you're interested, we can try, I can hook you up with that and sure you can try it out.
Yeah, that would be really cool. Thank you.
Yeah.
You know, Solomon and Abby, I did not email Edwina though and tell her that we were running way behind.
I got an email from her at 3:05.
Okay.
So I told her that she should be able to use the link now.
Great.
I can email her back if we don't see her in a little bit.
So let's see. We have 6, 7, 8, 9. We're up to 9. But I tell you what, why? Because of interest of time, I'm just going to launch this poll and we'll get started as soon as possible. I know more people are going to keep rolling in. Here comes a couple more now. They only had a few minutes in between, so I know I need to also run away for like a minute to come back. But I'll launch this poll, and I hope that everyone will be able to see it as they're logging in as well. So if you guys could answer this question, and it's a choose your own adventure today. Dr. Matela is being very flexible and will do majority rules. We'll be a true democracy. So we have, oh, it looks like the overview of sequencing seems to be winning. But that goes along with your original plan, so that maybe will make it easier.
That was.
Excellent. So here's the poll. We'll say, I don't think, there's no way it's going to catch up. We already have 8 of the 10 students say they want to hear about sequencing. And this isn't surprising because I think at least a handful of students are working on sequencing projects this year.
Cool.
Or with their mentors right now. So I think this will be of interest to them for sure.
So I think then here's my plan is I will go through that, that sequencing PowerPoint, and please stop me and ask questions as we go along. I know we only have half an hour today. And what I wanted to hear too then, just thinking, I'd love for everybody to be able to run through the Patrick analysis before tomorrow. I'll mention that I posted on Google Classroom the slideshow for tomorrow, but you know, just look at it if you're interested in previewing it beforehand, but I'll just walk through that slideshow tomorrow. So yeah, I guess, well, you know what, before I get into that, and I don't know the best way to have, unless everyone doesn't mind just answering, if you were able to do the Patrick analysis, or if not, where you had problems, so that once we go through the sequencing, I can maybe address issues and help you to get that completed.
Yeah. Everyone can just do the hand raising for this one.
I wasn't able to finish it. I got to the like variation analysis point, but I don't know what it was. I'd spent like a long time trying to put my files into the read directory or read library, but I couldn't. I don't know why.
The variation analysis. Oh,
That's good. Thanks, Alexandria, for
really
letting Dr. Matela know. Any issues you guys ran into, this would be really helpful for her to make sure everything is working.
I was able to get the variation analysis job to complete, but it was after 7 failed attempts. So I don't know what I did to make it work, but it worked eventually. So I don't know.
Excellent. Let's see. Oscar says, I couldn't access the files. Oh, that's is what I was emailing you before, Abby. I think it's an issue with my Mac because it kept crashing.
And I also have a Mac. I wasn't able to, I've tested it before and just, we don't, I mean, one thing, if you're trying, don't open the files. You don't need to, you won't be able to open them. But if that wasn't the issue, hopefully you could, I don't know if you're just having problems just moving them or getting them.
Anissa says she has a Mac and it worked fine. So could be individual machines as well. Angelina also says it. Well, actually, Angelina said same for me, but I'm not sure if she's referring to Oscar or Anissa. To Anissa. Okay, so that's good. So it could be an individual machine.
Yeah, maybe it's older. I'm not sure. I'm sorry. It's hard to know exactly. I'm trying to actually figure out, Alexandria, at what point. So you were able to get the files and load them, but then did you have an issue running?
I wasn't able to run it. It's probably like a stupid problem. I don't know if I'm like pressing the wrong spot, but I'm on the page now and it's like where it says read file and you're supposed to put forward in file 1 and like reverse in file 2. It won't let me upload the files. Like it won't let me select it won't let me select either of them.
Did you, oh, sorry.
No, go ahead. I was gonna say, you have to already upload them before that? Could that be the issue?
Yeah, I put them in a new folder, like I think the slideshow said.
I'm sorry. We can also, if anyone is having individual issues and they would really like to do this and they want to meet with me before class tomorrow, please just send me an email and I'd be happy to Zoom with you, and then we can walk through your specific issues, and I can help you and hopefully be able to help you to get it to work. So this is the first time we're like kind of sending this out on a massive scale for everyone to try. I will say, I can't remember who mentioned this, but someone had to rerun the analysis multiple times before it worked. I know that we a lot more data that we could have used, and I tried that, and there were plenty of them that didn't work, and I don't know why. These were the ones that did work for me. So if you are having an issue like that where you try it and your mutant for some reason doesn't work, I would just pick a different mutant if you want to do it and see if that one works. You know, they should work, but maybe I'm not sure why the program doesn't always work. Okay. Does anyone else have any other questions, just thinking about that and being able to actually run via the sequencing analysis before tomorrow?
So Oscar is asking, they're not sure if they have your email.
Oh, yes, it's on the PowerPoint. Wait,
okay.
I'll type it here too.
Yeah, this is always hard too. I mean, not only is it the first time piloting it, but you're not even giving them their own machines where you know that it works, which also makes everything incredibly more difficult.
You guys can use this email to email me. Yes, and I don't, and that's, yeah, sorry. As an aside, I would hope because it's online, it wouldn't matter with your machine, but maybe it does. I don't know. All right. If we're good with that, I will share and do the whole genome sequencing. Yeah, it's also at the beginning of all of our PowerPoint slides for you guys, that email address. Okay, yeah, and please stop me as I go through this if you have questions. All right, so, you know, we talked about before how these changes to the colony morphology are heritable. So at the end of an experiment, this is just an example of what the final day might look like, where you get a kind of, we always get a mix of different colony phenotypes, but some look like these ancestral smooth ones, but we can see that there are some differences like these big wrinkly spreaders. And so, so what we want to know, if what the bacteria are passing on to their offspring is their DNA, right? That's where you're getting that heritability. So if we grow one of these wrinkly colonies overnight and then plate them, we can see that all the offspring then look identical to their parents. And what we're then looking to find out is they look identical, but what's the genetic change that actually caused them to have this difference in phenotype from their ancestor? And we can do that by then sequencing. What we do is sequence the entire genome of each mutant to identify the evolved mutation. And I think that this is just a really cool thing now that we can do, because whole genome sequencing is pretty efficient. So we don't have to, you know, guess at where the mutation might lie and develop different primers. We can actually just look across the entire genome of our mutant, compare that genome then with these bioinformatic relator part to the ancestor, and see where did a mutation occur. So what's the first step in that? The first step, we got to get the DNA out of the bacteria. This is the procedure we use. We use a kit, this DNeasy mini prep kit. So maybe some of you guys have used this in your own lab work. So you just collect a sample of bacteria, and then you lyse or just open the cells up to release their DNA into liquid. And then you use a centrifuge, you know, it spins really fast to wash the liquid through a column where the DNA will actually bind to a membrane in this column. And now that the DNA is bound, you want to wash away all the other impurities that might have also attached to the column, and then really just get a buffer that will actually wash the DNA through. So in the end, at the bottom, you just have, you know, usually it's either a buffer or just water where you have a little pool of your sample of DNA. This is just a picture we actually have in our lab. This robotic system that automates that entire process called the Kaya Cube. I have never used it, but it's a cool way, you know, it's kind of neat that they have these whole neat machines that can just do everything. You don't have to sit there doing these processes by hand. Okay, so now we'll get into, so we have the DNA, but how do we find out that actual bacterial genome sequence? So this is just a brief overview describing the process for that. So we use that extracted and purified DNA, and how we figure out the DNA of the genome. Essentially what we do is cut the DNA from the whole genome into short fragments. And then we're gonna make lots of copies of each individual fragment. We'll then label these little groups with a fluorescent tag and then record information from that fluorescent signal with the computer. And all of these amplification, making more copies and the sequencing reactions are performed on this specialized glass slide called a flow cell. So this just gets into a little more detail of how that process works. So here, this is just representing little pieces of the DNA that have been randomly chopped up into tiny fragments. And then we'll attach them to this flow cell. So each fragment will just attach to a different position on the cell. And then once the fragments have attached, we use this reaction. It's called bridge polymerase chain reaction, or bridge PCR. You don't really, I think, need to focus too much on the details of that, other than to understand that in the end, what you want to get are clusters of identical fragments. So you're amplifying an individual fragment to make clusters that have multiple fragments that have where the DNA is identical there. And then the sequencing happens, and what we do there is kind of wash over these fluorescently tagged nucleotides, and then they'll attach to their complementary piece within one of these fragments, and then use a laser to excite the fluorescent signal, and then that information is captured by a camera. So the camera would kind of look across that whole cell, and it would know if it lights up in a certain area, that if you were adding, the fluorescently labeled T, that particular fragment had a T in that position. And then you just continue to do that, as a process to figure out what the sequences are in each of these clusters. So this is why using bridge PCR is necessary, really just to have a lot of copies so that you have enough of a signal to be able to be detected by that camera. This is just going, so you do kind of both forward and reverse strands. So essentially just read the DNA going one way and then flip it around and read it going the other way. The reason for that is just sometimes there are errors introduced during the sequencing process, depending on which strand you're looking at. So that's the point of also looking at the reverse strand. And so the genome is a little over 6.7 million base pairs. But when you're sequencing, you're only getting small chunks of that, right? So they're about 150 base pairs, I think, on average in size. And actually, maybe I will try to go a little bit quick through the next thing, because that kind of points out what coverage means, if that makes sense. But so really, in the end, you've done your Illumina sequencing. So you have the sequence of the genome, but all that information is just packaged in these short 150 base pair chunks, right? And the genome is millions of base pairs long. So what the bioinformatics program is going to do is take those tiny little chunks and piece them together to match them up into, in our case, a genome where the whole sequence is known already, so that it can line up each of these little pairs to reconstruct your mutant's genome and then identify any changes from the mutant to the ancestor. So that was kind of just a quick overview of how we do Illumina sequencing. But does anyone have questions right now?
Can you talk briefly about how you get like the DNA to go from like single stranded to double stranded? Is that applicable to this?
So how you're copying it?
Yeah, because I think I read in the PowerPoint, like you have the like reverse strand, but then it finds its complementary strand.
So how are you doing that? I'm trying to think of exactly what you're asking. So right, you'll have each single strand, and essentially they all attach because they have these little adapter sequences on the end that are identical, and that so they're randomly attached to the flow cell. And the polymerase, so doing PCR, we know what that's how we can prime to amplify that individual strand to make multiple copies, because we know, even though we don't know the sequence from the mutant's genome, we know the sequence of that adapter. So you can use primers to have that amplify that little chunk.
Okay, thank you.
Can you say again what was going on with the clusters and what they were made of? I don't think I like understood that.
Yeah, so the clusters are just, you start out like, so all those little pieces of DNA are stuck to different parts of the flow cell. And the clusters just mean you're doing PCR, so you're doing a reaction that makes exact copies of individual bits of DNA. And the cluster just means, so in a really close area, you'll have a bunch of identical copies of that DNA sequence fragment. And why we need that is because how you will read the sequencing is by emission of a fluorescent signal. And so if you only had one strand, that signal wouldn't be strong enough to detect. So that's why you make a bunch of copies, they're all real close together, and then the computer can detect like a flash right in that little area. Does that answer your question?
Yeah, that makes a lot more sense. Thank you.
Okay. I'll try to go because I know we just have like 10 minutes left, but let's go kind of quickly through the next slideshow, which is the actually doing this analysis with Patrick. So we're going to use Patrick's. We have the little strands, and we're going to then be able to match them up to a known reference sequence to see where a mutation happened in our genomes. We're using Patrick. There are a lot of bioinformatic analysis pipelines you can use. We're using that because it's easier than a lot of other things. You don't need to know coding, how to code that kind of coding language at all. And since it's available online, you don't need to download anything onto your computer to use it. So hopefully you were able to register for an account, and then you were assigned mutants, and you could see pictures of what they actually looked like. And then you go through to upload these forward and reverse sequences. And this is just creating them. Trying to see what the best parts to look at are. So yeah, so this is just like details on how to upload each of the sequences. And of course, again, please email me and we can go through if any of these areas were issues for you. Then we can like kind of share screen and I can walk you through to figure out why it was difficult to make this work. And then you wait and the files upload. And then you're gonna choose this variation analysis to actually run it. And then you add the reads to this paired read library, and then choose the parameters that you're going to use to both align the short reads to the reference genome, and also to this particular analysis program that will call where there are differences in your mutant's genome. I'm just gonna go back, because one thing I'll point out that can be an issue, if you type in for the target genome, Pseudomonas fluorescens SBW25, it, let me see if that's here. You have to pick, I think it says at some point, oh, maybe, sorry. There are two different ones you can pick from. And whatever it says in here, which I'm not finding right away, but you have to pick the exact one it says to use in the slideshow, or I think it will fail if you don't pick that one. I'm sorry, I'm not seeing where I wrote that. Okay, so when we're thinking about, so you're not able to see this with the Patrick tool, but if you had some kind of program that can look at sequences, you can visualize what the individual reads look like and how you would compare them to a reference genome. So what this shows you when you're looking at this slide is this is just one portion of the reference genome that's highlighted at the top. And that's all, this is like the DNA base pairs of that reference. And then each of these little lines are those read files, those sequenced reads that came out of the Illumina run. And so once you've finished, once the program has finished going through the alignment and the calling, this is what it's done. It's taken all those short reads and it's figured out how to match them to the reference genome to rebuild the mutant genome. So I don't know if that doesn't, if you have any questions now, let me know, because I don't know if that totally makes sense. I'll say again too, maybe looking at them, you know, there are these little breaks, like let's say here, but these are still different short read files. It's just, this program compresses them, so it's not like hundreds, you know, all the way down the page. So Freebase is this program that's gonna identify then how do the read sequences differ from the reference sequence we used. And so if we go back to that previous example, I think this is kind of a good visual of, in this case, Freebase is going to identify that there's a mutation in your genome here. And so, oops. So the reference has a G at that position, but after the sequencing was done and these short reads are aligned, we can see that in this particular mutant, that position has a T instead. So that's what it's doing. And when we think about kind of the errors and why you would need a program to figure out what's a real mutation compared to stuff that's just errors introduced either in amplification or alignment, you can see these so these changes in color, they're just indicating changes from the mapped reads to the reference. And you can see that happens all over, right? But those are just errors. So that Freebase program is saying, you know, it's kind of pulling out the errors and showing you the real mutations. Oh, here is actually that part. Okay, so this could be an important part why things might not work. When you type in that Pseudomonas fluorescens SBW25, you have to choose like this particular option as your reference. So it has to be like this REP, like that exact thing has to be used as the reference, or for some reason it fails, and I don't know why. And then you choose location for the output, and then you click on this to move your selected libraries over and then submit. And then you wait, and it should analyze your results. A lot of this next part is just how you would actually use either Excel or Google Sheets to kind of pull through the results. So you download this particular file. It's a tab separated value file. And you can put that on your computer or open it in Google Sheets. And then initially you'll get a whole long list of things that are potentially mutations. And what we're gonna do, there's also a list of details of what all the columns actually meant. The important, so first, then you're gonna pull through, or you're gonna kind of pull out the things that aren't important. So what we use as a reference is the published genome of Pseudomonas fluorescens, this particular strain. In our laboratory experiments, that ancestor has 3 mutations in comparison to that reference genome. So we know that these are just already happening in the background of those experiments. So we can immediately just delete those because they're not new to the mutants. And then we can sort to then also get rid of variants of low frequency. Why we want to do that is because we're analyzing clonal populations, so we expect them to be identical. All the bacteria should be identical. So we only want to see mutations that it's calling as being present in nearly all of the variants that were aligned to a particular region. And they could be lower than 100% just because of errors. So that's why we'll get rid of things under 80%. And then we also want to remove variants that it looked like maybe the fraction is high, but they're low quality. I think reasons for that, I'm not sure exactly what goes into the quality score here, but maybe it's low coverage in a particular area. So maybe there aren't very many reads that aligned to that area. That could be one reason, for example. So if there are not very many reads, but 80% of those reads are different, it'll give a high fraction, even though the coverage isn't very good. So we get rid of the low quality variants, and what you should come up then in the end is what mutations you found in any particular clone that you chose. So this is the example, and the output here just shows you that we actually found 2 mutations. So, and some of the additional mutants that you could have analyzed will also be double mutants, so they don't just have 1 mutation, but 2. So that's the 2 lines, 2 and 3 mean those are both individual mutations. And this one was actually in 2 different genes. So what is this output? What are we looking at here? So first, we can identify the position. So this is the position within Pseudomonas fluorescens' genome. So that's just pointing to exactly what point in that genome the mutation was found. You can see that in both cases here, there is just a single nucleotide change. So here the reference or the ancestor had a G and the mutant had a T. And then, so I guess one thing that I wanted to ask you guys was can anyone tell me what a codon is? Do you know what a codon, what does that mean?
It codes for an amino acid.
Yes. Yeah, so DNA is, the language of DNA, right, is nucleotides. But the language of proteins is kind of, it's amino acids, and those are indicated by these triplet DNA sequences that are called codons. So the code, so one thing to know is that codons can either code for different amino acids. You can have, the code is redundant because different triplets could code for the same amino acid. And then some of those codons, there are 3 that actually code for a stop. So to tell to say where to stop making this protein. That's the end. So in this case, we're actually getting a change that altered the codon sequence so that the ancestor had this GAA, but the evolved mutant had TAA instead of that position. So we can also, it's giving information about where that amino acid position occurred within the gene, and then how did it actually change the amino acid, right? So in this case, the ancestor had glutamic acid, and this is GLU is the shorthand for that, but the evolved mutant actually has a stop codon now at that position. So in the middle of the gene where there was a glutamic acid, now the gene has a premature stop and it's not being fully translated. So the other interesting information you can get from this, so locus tags are just unique identifiers for the gene. And you can use this then to learn more about your gene from a different, well, I think a good resource for doing that is using the Pseudomonas genome database. There's also some other information it might provide, like it might give you the gene name and maybe a little bit of information on its function. But sometimes, like in this case, it knew that this gene is already named and called WISPF, but the second one it doesn't give a name for. Just flip through. So navigating the genome database, you can just type in that pflu, and in this case, I'm doing the first one, that 1224, and it'll open up a page that looks like this, and you can click on the locus tag, and then it gives you a bunch of information on your gene. So this might be, if you're interested in stuff like this, a fun place to just kind of click around and see what kind of information you can find out about the mutants, or the mutations, I should say, in any particular mutant. You can click on sequences and actually look to kind of see how long the DNA sequence is and the protein sequence. Functional pathways are kind of interesting because it will tell you what it thinks, what's predicted for the protein to be, what's its predicted function. And another thing that's important for a lot of these, not a lot, but probably more than half of the mutants that you guys have to analyze, they don't give you a name for the gene that was mutated, either in the Patrick output or actually even if you came to the Pseudomonas database, it won't have a gene name listed. So that's where if you click on the ortholog group members, what that tells you is what is the same genes in other species. And clicking in there can then show you like, well, even though we didn't give it a name in Pseudomonas fluorescens, we know that this closely related species, Pseudomonas aeruginosa, that gene has been named. And so then you could write down the name of the gene, even though it's not provided in this initial look at fluorescens. So I know that was a lot of information. And I think what will be interesting tomorrow is we can kind of walk through what did those mutations mean, depending on what gene was mutated. What do we think that means for the bacteria and why it's allowing these bacteria to have increased biofilm production in comparison to their ancestors. Are there any, or what questions do you guys have before tomorrow? So the plan for tomorrow is try to get through and be able to do the analysis, because I think that's probably the most fun hands-on part of this whole module, given that it's virtual. If you have any issues, email me and we can set up a time, and then we can go through step by step to see if we can fix those issues for you. And tomorrow we'll just plan on walking through what did the mutations mean for the bacteria.
So Abby, there is one question in the chat. Angelina is asking, for the shared blank sheet that you shared with each of us, what are we supposed to write on it? She said she's already working on it. She wants to know if she is supposed to work with another student if they have the same mutation.
Sure, you can. Yes, you know what? It's in the what I thought probably is, and especially if you guys can all get this done, it might be interesting for you to share your results with other students. But if you go to the classroom and go to the Read Me First assignment overview.
Abby, do you want to share your screen, what you're looking at right now?
I can do that. So I'm just in the Google Classroom, that Read Me First document. So there's a lot of information in that, but at the bottom, just use the mutant assignment. What you should be able to report is the name of the clone you analyzed, and then what mutations you identified. So you can do that locus tag, that unique identifier, and then find the gene name. What was the nucleotide change that occurred? How did that change the codon? How did that change the amino acid sequence? And then maybe if you can figure it out on Pseudomonas, some idea of what might that gene be functioning to do. And I think, of course, if you want to work with other students, that's great, especially if you're having some difficulties. Even, we don't have to get through all 10 of those mutants. So yes, please feel free to work alongside somebody else to fill those out.
I have a quick question. For report the name of the clone you analyzed, it's just like the A_2019. Okay, I was just curious.
Yeah, that just lets me, I have like kind of a list of all the, what should have come out of the thing, and then I can match it up to say, yes, that was it. But, but I think if you guys can get through it, I've tried them a few times and I've gotten the same results every time. So hopefully it will work.
Excellent. Any last questions for Dr. Matela until tomorrow for our conclusion of the Evolving STEM module? Okay. Well, we'll see you guys tomorrow. So Solomon, what do we have tomorrow to preview? You're muted. Still muted.
Yeah, so tomorrow we have from 12 to 1, Nilgun Tazimir breast cancer overview. And then 3 to 4, the final Evolve STEM module.
Perfect. Sounds great. So I'll see everyone tomorrow.
Thank you.
Take care. So Abby, so sorry.